Today we will talk about the evidence for genetic material, the search for DNA being the genetic material. We studied what the genetic material is, then what are the evidences which prove that yes, it is the DNA which acts as a genetic material in living organisms. So two important experiments were performed which provide an evidence that DNA is the genetic material. And this first one was Griffith's transformation experiment. These two experiments provide a sort of evidence which suggested that it is the DNA which acts as a genetic material. One is Griffith's transformation experiment. Griffith's transformation experiment. Another is, second one is transduction experiment by Hershey and Chase. Transduction experiment by Hershey and Chase. Transduction experiment by Hershey and Chase. First we will discuss this Griffith's transformation experiment, which provides evidence that yes, it is the DNA which acts as a genetic material in living organisms. So first is, we'll discuss first one, that is Griffith's transformation experiment. Griffith's transformation experiment. This Griffith's transformation experiment, it is also known as Griffith effect. Griffith effect. Before discussing the details of this experiment, we have got what is the transformation. Transformation is simply a permanent change in the genetic constitution of an organism when that organism picks up the genes from the outside environment. So what do you mean by transformation? Transformation is the permanent change. Transformation is a permanent change. It is a permanent change in the genetic constitution of an organism. It is a permanent change in the genetic constitution of an organism by picking up the genes from the surrounding medium. By picking up genes from surrounding medium. So this is a concise definition of the transformation. Transformation is the permanent change in the genetic constitution of an organism when that organism picks up the foreign genes present in the surrounding environment. So what was this Griffith's experiment? We'll discuss this Griffith's experiment. Griffith did this experiment in 1928. 1928, Griffith made a transformation experiment. This Griffith, basically he was a medical officer by profession, and this Griffith was working on the pathogenicity of the bacterium. The bacterium was Streptococcus pneumonia. This Griffith, he was a medical officer by profession, and he was working on the pathogenicity, or he was working on the virulence of the bacterium. The bacterium was Streptococcus pneumonia. Streptococcus pneumonia. Streptococcus pneumonia. This Streptococcus pneumonia is also known as Diplococcus pneumonia, or it is also known as Diplococcus pneumonia, or simply pneumococcus. It is also called as pneumococcus. And this bacterium, it causes pneumonia in living organisms. It is responsible for what disease? It causes pneumonia in living organisms. It causes pneumonia in living organisms. This Griffith, he found that this Streptococcus pneumonia, or what is called as Diplococcus pneumonia, it has got two strains. It has got two strains. One is two strains, or we can say that two types. It has got two strains or two types. One is OR type, or strain. Another is called as S type, S strain. Probably it is S strain and OR strain. So it has got this bacterium Streptococcus pneumonia or Diplococcus pneumonia. It has got two strains. One is called as OR strain, another is called as S strain. This OR strain, it is the bacterium, it is a Streptococcus pneumonia strain, which is without capsule. That means there is no capsule, or we can say that outer covering which is present external to the cell wall of the bacterium. So this is what is called as OR strain. This OR strain, it simply means rough. OR means rough. That means this strain is not having capsule outside the cell wall. So it is, this OR strain, it is non-virulent. It does not cause disease. Non-virulent, or it does not cause disease. Then we have got S strain. S strain is simply what is called as S means smooth. S strain is simply having the mucus coat or capsule outside the cell wall. It is having the mucus coat or capsule outside the cell wall. And this because of the presence of mucus coat or capsule outside the cell wall, this strain of bacterium, its surface is smooth. That is why it is called as S strain. And this is virulent. That means it causes disease. So this OR strain, I told you that Streptococcus pneumonia, it has got two strains. One is OR strain, another is S strain. OR strain, that is rough strain, it is without the capsule coat. It is without the mucus coat outside the cell wall. Because of the absence of the capsule or mucus coat outside the cell wall, its surface is rough. That is why it is called as OR strain. And this OR strain, it does not cause the disease. It is non-pathogenic. It is non-virulent. Another strain, what is called as S strain. S means smooth. That means there is a capsule, there is a sort of mucus coat outside the cell wall of this S strain type of bacterium. As a result of that, its surface is smooth because of the presence of this capsule, and this is virulent. It causes the disease. So it would be like this. For example, this is bacterium. This is bacterium. This is plasmembrane. This is plasmembrane. Outside this is cell wall. So this is cell wall. This is plasmembrane. That means there is no capsule or there is no mucus coat outside the cell wall, so its surface is rough. So here we have rough surface. That is why this type of bacterium is called as or strain. Well, as in this case, in this type of Streptococcus pneumonia, we have, this is nucleoid, genetic material nucleoid. This is plasmembrane. Obviously this is cell wall outside the plasmembrane, and outside this cell wall there is a mucus coat or capsule. That means there is a mucus coat, capsule outside the cell wall. So this is capsule, this is cell wall, and this is plasmembrane. This is nucleoid, genetic material of the bacterium. Obviously the surface of this bacterium is smooth. That is why it is called as what? It is called as S type of the Streptococcus pneumonia. Now he experimented this Griffith in 1928. He experimented with this Streptococcus pneumonium, and he injected these two different types of Streptococcus pneumonia, two different strains of Streptococcus pneumonia into the mouse, and then observed the results. So it would be like this. The experiment is like this. He injected these S type of bacterium into the mouse. He injected R type of bacterium into the mouse, and then observed the results. He performed his experiment in 4 steps. We have case first in 4 cases. Our first step, that means we have live R type of bacterium in the culture medium, and he injected live R type of bacterium. This is live, living R type of bacterium into the mouse. He injected live or strain of bacterium, live rough type of bacterium into the mouse. And what he observed, he introduced the live or type of bacterium into the mouse. The mouse survived. That means when he introduced the living rough type of bacterium, rough type of Streptococcus pneumonia into the mouse survived. Then another step is he injected, this is S type of bacterium, that means a smooth type of bacterium, that is S strain of bacterium, smooth strain of bacterium, which is virulent. He injected into the mouse. He injected into the mouse. He injected it into the mouse. What happened to the mouse? Because of this is virulent. This is virulent. This is virulent strain. The mouse died. Mouse died. The mouse died. In this case, the mouse developed the pneumonia and ultimately the mouse died in case second. Then case third, he injected heat killer. This is S-type bacterium, but here it is heat killer S-type bacterium. This is heat killer. This is heat, and it's called as heat killer S-type. He injected the heat killer S-type bacterium into the mouse. He injected the heat killer S-type bacterium into the mouse. What happened to the mouse? The mouse survived. Mouse survived. Now, in the fourth case, he introduced a mixture of heat killer S-type bacterium and a live R-type bacterium into the mouse. What he observed? He introduced, this is mixture of mixture of heat killed plus live or strain. This is the mixture of heat killed S strain of bacterium plus live or strain type of bacterium into the mouse. Into the mouse. What happened to the mouse? Mouse died. See, although separately when the heat killed S strain was injected into the mouse survived. Separately when the live or strain was injected into the mouse survived. But when he mixed the living or types of bacterium and this heat killed S strain of bacterium, and then collectively in a mixture of these two were inoculated into the mouse died. And to his surprise, and when the autopsy of this mouse was done, the living S strain of bacterium were recovered, and living S strain of bacterium were recovered after the autopsy of this dead mouse. Now the question arises that these two separately they were unable to cause the disease, but when they were mixed together they caused the disease, they caused the pneumonia, which resulted in the death of the mouse. So Griffith said that a transforming substance has released by the heat killer S strain of bacterium, which was absorbed by the live or strain of bacterium. As a result of that, this live or strain of bacterium got transformed into the S type of bacterium. So what he said that this or strain of bacterium has got converted into S strain. How it has got converted into S strain? Because it has absorbed a transforming substance. This transforming substance has released by the heat killed S strain of bacterium. As a result of that, this R strain has got transformed, it has got changed into the S strain. And this Griffith, he failed to explain the biochemical nature of this transforming substance. So we can say that Griffith only said that it is a transforming substance which was absorbed by the R strain of bacterium, and that living R strain of bacterium changed the R strain, transformed the R strain into the S strain. That is why we obtained S strain type of bacterium from the dead mouse. So Griffith failed to explain the transforming principle or transforming substance. Transforming principle or transforming substance. He was unable to explain what this transforming substance is. What is this substance which was responsible for the change of R strain of bacterium into the S strain of bacterium? What substance changed the R strain of bacterium into the S strain of bacterium? Then in 1943, three scientists they did the biochemical analysis of this transforming substance. They were Avery, MacLeod and McCarthy. Avery, MacLeod and McCarthy. Avery, MacLeod and McCarthy. They did the biochemical analysis of transforming substance. This Avery, MacLeod and McCarthy, they did the biochemical analysis of this transforming substance. What I mean to say is that they analyzed the biochemical nature of this transforming substance. What they did, they took the S type of bacterium, S type of Streptococcus pneumonia or S type of Diplococcus pneumonia, which is responsible for causing the disease pneumonia. S means smooth, it is virulent strain. And they fractionated this S type of bacterium into three components in order to study the biochemical nature of this transforming substance, transforming principle. What they did, they simply fractionated the S type of bacterium into three components. One is proteins, another is RNA, then RNA carbohydrates, and DNA. That means they fractionated this S type of bacterium into four components: proteins, RNA, carbohydrates, and DNA. And then these were separately added to the living R type bacterium. So we have culture medium where we have what? We have R type of bacterium, R type of bacterium, R type of bacterium. So R strain was present in a culture medium, and then protein of S type, S strain of bacterium added. Protein of S strain added into the culture medium having R strain of bacterium. So what they found? They found no transformation. No transformation. That is, R strain doesn't got transformed, doesn't got changed into the S strain. So it means that protein is not the transforming substance, because proteins have not transformed, proteins have not changed the R strain of bacterium into the S strain. So protein is not the transforming substance, transforming principle. Then next, same, we have culture medium having R type of bacterium, R type of bacterium, living R type of bacterium, R strain of bacterium, and RNA was added. RNA of S strain was added. S strain was added to this culture medium having R strain of bacterium. So R strain of bacterium, non-viral strain of bacterium, living R strain of bacterium. This is living R strain of bacterium, living R strain of bacterium. So what they found? They found no transformation. That means R type of bacterium or strain of bacteria had not got transformed into the S type. So no transformation. It means that RNA is not the transforming principle. RNA is not the transforming principle, or RNA is not the transforming substance. Then same living or strain of bacterium, living or strain of bacterium, and carbohydrate of S type, S strain was introduced in the culture medium having R type of bacterium. What they found? They found no transformation. No transformation. That means R strain has not transformed, has not changed into the S strain when the carbohydrate of S strain was added to the culture medium containing the live R strain bacterium. Then another case, we have R type of bacterium or type of bacterium. Then this is DNA of S strain. DNA of S strain was added to the culture medium having R strain bacterium. They found transformation. That means they found transformation. That means this changed R strain of bacterium into the, that means R strain changed into the S strain, or strain changed into the S strain. So we can say that DNA is the transforming substance. DNA transforming substance. DNA is the transforming substance. It is the DNA which has transformed the living R type of bacterium into the S type of bacterium. So this clearly shows that it is the DNA of the smooth type of bacterium which has transformed the R type of bacterium into the S type of bacterium. So they also used it when they also treated this extract, this S type extract was treated with DNA. It was treated with DNases. When S-type extract was treated with DNases, DNases are the enzymes which destroy the DNA. It was found that no transformation. No transformation. Because the DNA has got destroyed, so there would be no transformation. So same is the case. S-type extract, it was treated with what? It was treated with RNases. RNases. That means RNases are the enzymes which cleave, which destroy RNA. So they found transformation occurred. Transformation occurred because DNA was present, that was intact. Therefore, the transformation took place. Also, S-type extract treated with what? It was treated with proteases. Proteases are transformation took place. Proteases are enzymes that destroy the proteins. Proteins are destroyed. It has caused no harm to DNA. Still transformation took place. RNA was destroyed. Still transformation took place because DNA was intact. DNA caused the transformation of R-type bacterium into the S-type bacterium. So we can say that how we can explain this transformation. This is, for example, this is R type of bacterium. R type of bacterium. This is DNA of S type. DNA of S type. So this DNA of S type was absorbed by the R strain of bacterium. So we can say that transformation is the uptake of foreign DNA by the cell. So what do you mean by transformation? Transformation is simply uptake of foreign DNA by the cell. This is called as transformation is simply the uptake of foreign DNA by the cell. Or we can also explain the transformation is the permanent change in the genetic material as well as in the phenotype of an organism by taking the DNA from the outside environment. So this is what is called as transformation. Transformation is defined as the permanent change in the genetic constitution, in the genetic material as well as in the phenotype of an organism by picking up genes from the surrounding environment. This is what is called as transformation. And then now the question arises that we have got a question that why R strain is non-virulent and S strain is virulent. We have a question that why R strain is non-virulent and S strain is virulent. That means R strain no disease and S strain it causes disease, it causes pneumonia in living organisms. Why it is so? See, this I told you that in case of S strain, Streptococcus pneumonia or S strain Diplococcus pneumonia, there is a capsule. There is a capsule. This is capsule, for example, this is plasma membrane. Outside the plasma membrane is cell wall. So we can say that this capsule, this is capsule, this capsule is present outside the cell wall. So this capsule is simply the mucus coat, and this capsule, this is a mucus coat or extra covering which is present outside the cell wall. This capsule acts as a, you can say that it is just a blood-proof, it is sort of blood-proof jacket, and this capsule, it resists the immunity attack of the host. For example, I will explain like this. If this is a, for example, this is a S type of bacterium. It is having the capsule mucus coat outside the cell wall. When it enters into the host, obviously the host will activate its immune system and it will form the antibodies. Those antibodies will start acting on this bacterium, but these antibodies would be ineffective because of the fact that this bacterium has got an extra covering, extra jacket in the form of capsule, and this bacterium will get survived, and in turn this bacterium will cause the disease. In turn this bacterium S type will cause the disease in the host, and that host will develop the pneumonia and that will die. So this is why this S strain type of bacterium is virulent or pathogenic. Whereas in case of OR strain, we have got OR strain, this OR strain, no capsule. That means there is no capsule wall, there is no mucus coat outside the cell wall. As a result of that, when this OR strain enters into the host, this host immune system will get activated and they will attack this OR strain type of bacterium, and this OR strain will get killed. So this because why it will get killed? Because it does not have an extra capsule, extra mucus coat which will protect this bacterium, and ultimately it will get killed and it will not cause the disease. Obviously it would not cause the disease and the host will survive. So we say that OR strain is non-pathogenic. It is non-pathogen, that it does not cause the disease. Now in case of I told you that this heat killed S strain plus living OR strain, when injected into the mouse, when mouse died, and we also obtained what? We also obtained S strain of bacterium from the dead mouse. What is the reason behind it? See, we have heat killed S strain. When heat killed S strain was introduced in the mouse alone, separately, when heat killed S strain was introduced in the mouse survived. Also when living OR strain were introduced into the mouse survived. But when these were mixed together and then were introduced into the mouse died. See what happens. This heat it has destroyed the capsule. It has destroyed the capsule of this OR strain. As a result of that, this heat has destroyed the capsule of S strain. As a result of that, S strain of bacterium is unable to cause the disease because its capsule mucus coat has been destroyed. It would be easily attacked by the antibodies of the host. But its DNA has survived. But DNA is intact because heat has no effect on the DNA. DNA is still there. It is intact there. And when we have just mixed this heat killed S strain with the living OR strain, this DNA got introduced into the living OR strain. As a result of that, living OR strain got transformed into the S strain, and this S strain of bacterium were recovered from the dead mouse. I hope that you have understood. What I mean to say is that this heat, it has destroyed the capsule, but the DNA is still intact. There is no effect of heat on the DNA. And when we have mixed this heat killer S strain of bacterium with the living R strain of bacterium, this heat killed S strain, its DNA has got introduced into the living R strain. As a result of that, living R strain has got transformed into the S strain. As a result of that, it causes the disease in the mouse, and ultimately mouse died. So this was the reason that why heat killer S strain and living R strain together have caused the death in the mouse. So we discussed about what is transformation experiment, what is Griffith effect. This Griffith transformation experiment is also called as Griffith effect. Then we discussed R strain type of bacterium, S strain type of bacterium. Then we discussed about the biochemical analysis of this transforming substance, and ultimately this experiment, they provide a proof. It is a sort of evidence which suggests that it is the DNA which acts as a genetic material. It is the DNA which was picked up by the R strain of bacterium, and that R strain of bacterium got transformed, got changed into the S strain of bacterium. So that is why transformation definition was given, that it is the transformation is defined as the permanent change in the genetic material, genetic constitution, as well as in the phenotype of an organism by picking up the genes, by picking up the DNA from the surrounding medium. Thank you so much.